Warm welcome to all to my channel World of Zoology. This is our third application of the animal biotechnology, which is nothing but gene therapy, ex vivo as well as in vivo gene therapy. Ex vivo and in vivo gene therapy with the examples from each: severe combined immunodeficiency syndrome, that is nothing but the SCID, and cystic fibrosis. Therapeutic measures for management of the genetic diseases range from supportive care dedicated to elevating pain and enhancing the quality of life to cure which eliminates all clinical features of the said disease. Most of the known genetic diseases are the ones that are due to inherited inborn errors of the metabolism, and to lesser extent the ones that are due to defective structural proteins. Although the various strategies involved in treating the genetic diseases, gene therapy has emerged as a consequence of recombinant DNA technology. Here, therapy means curative treatment. With the help of the genetic engineering technique, a large number of enzymes and structural proteins can now be made available for therapeutic use in diseases like severe combined immunodeficiency disorder, the SCID, and cystic fibrosis, insulin dependent diabetes, etc. This method involves the introduction and expression of genes at their biological sites of action in humans, which replace the respective defective genes that are responsible for a particular genetic disease. Theoretically, the gene therapy should provide a persistent in vivo treatment, that is, inside the body, either in the tissue that is primarily affected by gene mutation or in deficient cells that acquired a recombinant protein from the distant cells which release the therapeutic protein into the circulatory system. Gene therapy is essentially classified into two: one as ex vivo and second one in vivo gene therapy, depending on modality of the transfer of the normal gene which replaces the defective gene. First, we'll consider ex vivo gene therapy. It is performed where the genetic alteration of patient's target target cells are carried out in culture medium, that is outside the body, and then these cells after correction are transfused back into the patient's body. Targeted cells from the patient are infected with recombinant virus vector containing desired therapeutic gene. These modified cells are then reintroduced into the patient's body, where they produce the needed protein that is encoded by inserted gene. Example of ex vivo or ex-vivo gene therapy. This procedure has been used to treat severe combined immunodeficiency syndrome, that is SCID. People with this disease are susceptible to infection because their white blood cells, WBCs, do not produce an enzyme, adenosine deaminase, that is ADA, needed by their immune systems. Hence, SCID is also called ADA deficiency or ADA SCID. It is actually an autosomal recessive metabolic disorder. The enzyme ADA is enclosed by a gene on autosome. The chromosome number 20 and the defective ADA gene, one inherited from each parent, has to be in its homozygous condition to express itself. If it is in homozygous condition, then expression takes place. If it is in heterozygous condition, then it serves as a carrier. Here, the example is shown, that is for gene therapy for SCID, where the bacterium carrying plasmid with clone normal human ADA gene combines with the genetically disabled retrovirus. Clone ADA gene get incorporated into the virus. Then T cells with disabled ADA gene are isolated from the young SCID victim, and retrovirus infect these T cells, transfer the normal copy of the ADA gene to these cells. These cells are then grown in a culture containing neomycin to ensure ADA gene is active. And then these genetically modified cells are re-implanted back into the patient and that produce ADA in normal fashion. Therefore, the diagrammatic illustration explains the methodology involved in ex vivo gene therapy for severe combined immune deficiency syndrome, that is SCID. The similar methodology with certain modifications can be used for treating many human diseases of similar nature. For example, lymphocyte associated cancers and other cancers where lymphokine activated killer cells, that is, LAC cells, are genetically modified to treat cancers of various organs. Severe combined immune deficiency syndrome is a fatal genetic disorder. One form of SCID is caused by a defect in a gene producing enzyme adenosine deaminase. The lack of enzyme causes destruction of lymphocytes of the human immune system. In the gene therapy, peripheral blood lymphocytes, that is PBL cells, are removed from young SCID victim, and these cells are then cultured in vitro in presence of OKT3 monoclonal antibodies and phytohemagglutinin, which stimulate lymphocyte to undergo excessive proliferation or growth. The cultured lymphocytes are then mixed with genetically engineered non-virulent retrovirus containing normal copy of ADA gene and marker gene for resistance to neomycin. The retrovirus transfect lymphocytes with ADA gene and neomycin resistance gene. Only the modified lymphocyte with neomycin resistant gene grow in a medium containing neomycin. They are allowed to proliferate up to 10 raised to 6 cells per ml and then transfuse back to the patient through intravenous route. Since the patient's own cells are genetically modified in vitro, that is outside the body, and given back to the patient, this technique is known as ex vivo gene therapy or ex vivo method. The lack of enzyme ADA results in the accumulation of 2-deoxyadenosine triphosphate, that is dATP, which in turn leads to build up of dATP in all cells, which inhibits ribonucleotide reductase and prevent DNA synthesis, and hence cells are unable to divide. Since developing T lymphocytes and B lymphocytes are some of the most mitotically active cells, they are highly susceptible to this condition. Blood lymphocyte, that is T lymphocytes, having normal ADA gene are cultured in vivo, that is inside the body. The DNA is isolated from these cells, and ADA gene is cut by a specific restriction enzyme which is then inserted into LASN, that is erythroviral vector adenovirus. Such infected vectors subsequently express human ADA and cDNA, which is a recombinant DNA. These are then cultured with bone marrow cells or with peripheral blood lymphocyte T cells of the patient suffering from the SCID, which is considered previously in this lecture, thus getting transfected with viral vector as they incorporate the ADA gene. Thus, the defective cells get corrected. Such cells are further enriched by in vitro amplification and then infused in the patient. The toxic level of dATP now gets lower because of the presence of the circulating lymphocytes possessing normal gene encoding enzyme ADA. On 14 September 1990, Kenneth Kluwer, Michael Bliss, and French Anderson initiated a clinical trial on ADA deficient children that resulted in successful gene therapy for SCID syndrome. Therefore, French Anderson is known as the father of the gene therapy. Now we'll consider the next, that is in vivo gene therapy. Previous gene therapy is ex-vivo gene therapy, where patients' own cells are modified in vitro and then given back into the body of the patient or reintroduced into the body of the patient. Here in this therapy, it involves introduction of the therapeutic gene directly into the patient's body. Hence, the DNA is introduced by cell-specific direct injection into the tissue in need. DNA in the form of plasmid vector is introduced by dermal vaccination. Once inside the body and in contact with the specifically targeted cells, inserted DNA is incorporated into the cells of the tissue where it encodes the production of the required protein. That means at the particular site, the modification takes place. Example of the in vivo gene therapy, which is cystic fibrosis. Cystic fibrosis is an autosomal recessive disorder. It is caused by the presence of mutation in both copies of gene for the protein cystic fibrosis transmembrane conductance regulator, that is CFTR. This gene was mapped by Lap-Chizwi, located on the upper arm of the chromosome number 7. The mutated gene results in the defective cell membrane transport protein. The heterozygous individual with single normal allele are carriers and are otherwise mostly normal. It is also known as mucoviscidosis, affecting mostly the lungs but also the pancreas, liver, kidneys, and intestine. The long-term effects include difficulty in breathing and coughing or sputum as a result of frequent lung infection due to mucoviscidosis. CFTR is involved in the production of the sweat, digestive fluids, and mucus, and when not functional, usually thin secretion becomes thick. This immediately results in buildup of thick mucus in lungs and body's airways. This mucus becomes the site of bacterial infection that is difficult to control with the help of even antibiotics. This prevents normal organs functioning by blocking the ducts and thereby aggravates the effect of cystic fibrosis. The primary effect of faulty CFTR gene is an alteration of protein that normally act as a chloride channel. As consequence of disruption of the proper flow of chloride ions into and out of the cells, mucus get accumulated in the ducts of the several organs, especially the lung and the pancreas. The name cystic fibrosis refers to the characteristic fibrosis and cyst that forms within the pancreas due to lack of the transportation of the chloride channel across the membrane, which is shown here in this diagram, where the excessively salty sweating by the skin is shown. Then the pancreas, that is exocrine pancreatic insufficiency, fat soluble vitamin malabsorption, cystic fibrosis related diabetes that results. Then also with respect to the osteoporosis, then hypertrophic pulmonary osteotrophy, also with respect to the intestinal obstruction in the syndrome, also with respect to the gallbladder and the liver hepatic steatosis, and the last one, that is small airways obstruction, recurrent respiratory infection that occurs in the patient and that cannot be controlled by the antibiotic, is shown in this diagram of the cystic fibrosis. Though the gene therapy for cystic fibrosis is not yet reliable, sure, there have been more than one recombinant technology approaches for the gene therapy of the cystic fibrosis, one of which is using adenovirus as a vector to slip the CFTR gene into the patient's lung cells. The adenovirus is an excellent choice as a vector because of the comparative ease of the means of inserting its DNA into the human lung cells. Further, these viruses produce high titer of their stock. Large cDNA inserts can be accommodated by these vectors, facilitating the insertion of the complete gene sequences. One of the disadvantages of using viruses as the vector is that these may be invaded by the immune system of the host, and hence the technique may not always turn out to be fruitful. That means it may cause infection invaded by the immune system of the host, and therefore the due to the attack of the immune system, the technique may not be fruitful sometimes. Thus, the alternative therapy for cystic fibrosis is the use of liposomes, that is the another vector, which is a tiny bubble of lipid or fats to deliver the therapeutic gene. Lipid vectors appear to carry fewer risks than viruses, and they are less likely to be viewed as the invader by the immune system of the human body. Most of such available formulations target the nostrils or lungs. These liposomes are sprayed into the nose and mouth of the cystic fibrosis patients as an aerosol or administered into the airways. Liposomes fuse with the lipids in the cell membrane of the tracheal cells, releasing normal CFTR gene into the cytoplasm of the cell. As you know that the cell membranes are lipoprotein in nature, the addition of this particular gene into the cell is easier. The normal CFTR gene is copied into the mRNA and the normal protein is translated from that mRNA. The normal CFTR protein enters the cell membrane and start to transport chloride ions out of cell, thereby thinning the mucus and elevating the CF symptoms, because holding the chloride inside the cell membrane within the cell will absorb more and more water due to the osmosis, and therefore removal of the chloride ion out of cell is essential, and that results in the normal CFTR protein enters the cell. As it enters the cell membrane, it starts to transport chloride ions out of the cell, and that results in thinning the mucous membrane of the particular organ. Thank you for watching this video with the two gene therapies, that is ex-vivo gene therapy and in-vivo gene therapy. In ex-vivo gene therapy, the correction is carried out in vitro, and then the corrected copy of gene is transported back into the body of the patient. Whereas in vivo gene therapy, at the site of the particular action, the normal copy of the gene is introduced with the help of the vehicle or the vectors such as retrovirus, or it may be liposomes, etc. Therefore, uh, thanks for watching this video of the gene therapy with the two techniques, in vivo and ex vivo gene therapy. If you like the video, do like, share this video for the benefit of the maximum students, and subscribe the channel World of Zoology for the further notification of the next video. Thank you.